studying histology. I will cover this during the first half of the session, and then after a break, I will cover cell membrane, the first lecture on cytology. So let's begin now on what histology is and how we study it. Are you following? Do you listen to me? ይሰማችኋል? Okay, I will go ahead. So you know, we have histology somewhere in between of our body's organization, which starts, if we begin from atomic level, we have atoms forming molecules. Molecules will give us macromolecules. The macromolecules would give us organelles found in a cell. Organelles are small machineries such as mitochondria, lysosomes, which are found within cells. In turn, several cells, along with non-cellular structures, give us tissues. It is the collection of tissues that make organs, and organs give us organ system, which in turn would give us the organism. So as you could see here, we have somewhere in between tissues. Histology is literally the study of tissues. So our topic revolves around here, around the tissues, but as you know, we also have to see cells and also organs. So that's what I'm going to lecture. I'm sorry. How many pictures, how many sharing do you see? Is it only one or multiple? ናትናኤል አንድ ፓወርፖይንት ብቻ ነው ሼር ላደርግ የፈለኩት፣ ግን እዚህ ላይ ብዙ እያሳየህ ነው ይለኛል። ብዙ ነገር ነው እያሳየው ያለሁት።
እኛ ጋር አንድ ነው የሚታየው።
ኦኬ፣ ከሌላ ጋር ሚክስ አፕ አድርጎ ነው መሰለኝ። ምን ይለኛል? ቱ ሜኒ ኦቨርላፒንግ ዊንዶውስ ይላል። ዩ አር ሼሪንግ መልቲፕል ዊንዶውስ ይላል። እንግዲህ የት እንደሄደ አላውቅም። ሜይቢ አይ ሃቭ ቱ ቢጊን አጌን ኦር ዋት ዱ አይ ዱ?
አይ ቲንክ እንደዛ ይሻላል።
ኦኬ፣
እኛ ጋርም የሆነ ነገር እየመጣብን ነው።
ኦኬ፣ ሌት ሚ ስቶፕ ሼሪንግ። ስቶፕ አድርጌዋለሁ። አሁን ምንም አይታያችሁም?
ምንም አይታየንም።
ኦኬ፣ ቪዲዮዬንም አትፈልጉትም? እሱንም ላጠፋ ነው። እሱም ይሆናል የሚበጠብጠኝ። ስቶፕ ቪዲዮ። ኦኬ፣ ናው I begin to share now. Okay. Okay. So here it is. My second slide is to show you or remind you the units of measurement we frequently use in histology. Oh, sorry, that's not the slide I wanted. Before that, let me tell you about a little bit about the divisions of anatomy. You know, histology itself is a branch of anatomy where we have under anatomy 3 divisions. The first one is macroscopic anatomy as gross anatomy. Then we have microscopic anatomy, which I'm speaking today, which includes cytology and histology. And we also have developmental anatomy, which is embryology. So cytology and histology are branches of anatomy which we study by the use of microscope. That's what we are dealing with today. The reason why we are supposed to study histology and cytology is because This would enable us to know and understand the three-dimensional tissue structures. Look at this orange. The orange could be representing an organ. When you look at this orange from outside, you don't see what it is made up of, or what we have inside is not visible. But by cutting, you can see inside. By looking at such inside sectioned part of this orange, you know and you can understand where the orange juice is produced. And you can see it is detailed. You can see it is three-dimensional microscopic structures. So that's what histology does. In histology, we cut, we slice our organs so that by looking at it under the microscope, we can able to understand the three-dimensional structure of our tissues. So that's the first objective of studying histology. The second reason of studying histology is it enables. Are you okay? Are you? የተናገራችሁ ነው ናትናኤል?
አቤት።
ኦኬ ሰላም ነው?
ሁሉ ሰላም ነው።
ኦኬ ፕሊዝ ነው። So the second objective of studying histology is to enable us understand the relationship between tissue structure and the function. The histology, therefore, is a base for understanding physiology. It's also important to enable us know the base for disease, for histopathology. When our tissue, our organ is diseased, it is detailed structures would be damaged. So in order to know this, for purpose of treating and curing, you have to know the detail of an organ. You know this by studying histology and cytology. So those are the basic reasons we are studying histology. Then, as I said earlier, we are going to deal with small detailed structures. For knowing the detailed structures, we have to be reminded the units of measurement we use for such small objects. These are usually around millimeter, micrometer, and nanometer. I would like to remind you that a millimeter is 1/1000th of a meter. In other words, 1 meter is made up of, or has in it, 1000 millimeter. So you have to think according to this. Similarly, we have micrometer, frequently used in histology. It means 1 micrometer is 1/1,000,000th of a meter. You have to imagine the dimension of a meter. So out of this 1 meter, 1 micrometer is 1/1,000,000th. When it goes to nanometer, it is in billions, because 1 meter is 1 billion nanometer. So you have to remember this unit of measurements we frequently use in histology. Like, for example, you study histology under light microscopy, you study cytology under electron microscopy. For light microscopy, our tissue has to be sliced down to a thickness of about 5 to 10 micrometer. It's very thin. When it comes to electron microscopy, we have to get it sliced down to 40 to 70 nanometer. So that's what we are using. Unfortunately, we don't have to use electron microscopy here in this country because we don't even have it, but we use photographs produced from outside. Otherwise, we use light microscopy. And as I said, for light microscopy, the tissue has to be sliced down to 5 to 10 micrometer. Think of our tissue is just nothing like meat, we get from butchery. That meat is just like our tissue. But now question arises. I'm saying to study this meat, that meat, our tissue, has to be sliced down to about 10 micrometer. But there is a question now, how is it possible to get this much, such very thin slice of the tissue? It is not possible to slice it in an ordinary manner. Not only this, the tissue would decay. The tissue would decay immediately because of self-digestion, autolysis, as well as putrefaction, bacterial digestion. Our tissue decays. That's what we know from our day-to-day activities, experience. But the histology deals with such slices which are made permanently. So for all this, we need to have a method of preparation of our tissue for microscopy and a method of studying histology. That's what I'm now going to speak. Because this is all known as tissue processing for microscopy. For this purpose, we have to first, immediately, we get our tissue, we have to prevent decomposition. Decaying has to be prevented. The second important thing, once the tissue is prevented from decaying, we have to have a manner of slicing our tissue down to micrometer thickness for light microscopy. When it is for electron microscopy, it has to be sliced down to nanometer. So this is only possible if we make our tissue optimally soft to be cut, but also rigid to be sliced. You can now compare or know or be reminded why our tissue, our day-to-day meat or food we have is decaying, as well as we are not able to slice, for example, from meat, very thin section. This is because of the following. One, the reason why decaying takes place is because of putrefaction and autolysis, and these are able to be prevented by a process known as fixation. So in tissue processing, the first step one needs to do is fixation. Once the tissue is fixed, for fixation, we have different types of fixation. The simplest ways of fixing, like we have in our day-to-day experience, is by physical fixation. We change temperature. Like, for example, we cook. Like, for example, we put our food in a refrigerator. That means we reduce the temperature. So one method of preventing decomposition is by changing temperature. In histology also we can do that, but that's not the optimum way of fixation. Instead, we have a fixation of tissues by chemical. Like, for example, look at this slide. This is a tissue, this brownish outline. By the way, do you see the pointer? Are you with me? Okay, I see now. Thank you. I've seen now you are following. So you see, here the tissue is cut by scalpel, and the tissue is now a few millimeter by millimeter, you can now fix it by chemical. One chemical widely used is formalin. You know formalin fixes. So this type of fixation is by immersion. You can immerse the few millimeter by millimeter tissue section into a liquid fixative, such as, for example, formalin. Fixation can also be done by a method known as perfusion. When the object you are going to fix, prevent it from decaying, is big, or maybe you want it to be fixed immediately, instead of immersion, you may inject the chemical through its blood vascular system. Like, for example, in gross anatomy, cadavers are prepared. The cadaver is just nothing but a dead body. The dead body cannot be fixed by immersion. Instead, what they do is they are fixing by a method known as perfusion. But for purpose of histology, simple way of fixation is by immersion, like this one shown here. And after some hours, or maybe overnight, your tissue would be permanently prevented from decaying. And now we can say our tissue is fixed. So we have now overcome the first problem in our day-to-day activity, which is decomposition. But the second problem is to slice our tissue. For getting this much slice, it is very difficult unless we have some method of preparation. Like, for example, I can ask you now one or two questions to think of it. Like, for example, if I ask you, is it possible to get much thinner section from a meat or potato? You would answer it is potato. Or from carrots or meat, you again tell me it is carrots that can be very sliced, very thin. Or if I again ask you between a potato and a plastic, you would answer to me plastic would be very sliced, very thinner than the potato, which in turn is able to be sliced very thin when you compare it with a flesh. The reason why potato can be sliced very thin compared with meat is because it is rigid, and yet it is still soft. The reason why plastic can be sliced very much thinner than potato is because it's much rigid, and yet it is able to be sliced, provided that our knife is able to cut it. So it is still soft, but it is rigid. So what we therefore need now to slice our tissue for preparation of slides for microscopy is to make our tissue rigid enough to be manipulated, yet it has to be cut because it has to be soft. And this would be done by replacing the water we have in our tissue by some agents that would make our tissue rigid and also remain soft. First of all, the reason why our tissue is not rigid enough to be manipulated is because it's full of water. We have in our body large amount of water. So we have to remove the water and then replace it by another media. So that's what I'm going to show you in a series of steps of tissue processing. In another method, you can also convert the water we have in our tissue into ice. Like, for example, in frozen tissue preparation, we can also slice very thinner section if we freeze our tissue, meaning by converting the water we have in our tissue into ice. This is also a method used in histology But this method is not for teaching purposes because it doesn't give us very thin section, as well as it is not a way of preparing permanent tissue slides. It is rather used for purpose of researches. So we need to have another method, for example, for teaching purposes or for studying tissue morphology. So in this type of processes, as I said earlier, we have to replace what are present in our tissue by another agents. The agent we use include paraffin wax. Paraffin wax is just nothing like a candle. I didn't ask you earlier. If I asked you whether we can or cannot get thin section from candle, you would have answered candle is even optimum and it is more suitable to be sliced than carrot or potato. So what we do for light microscopy is replacing what are present in our tissue by wax, like a candle. And you can now think candle can be sliced. For electron microscopy, we replace the water present in our tissue by plastic resin. And you know that plastic is able to be sliced very thin. But there is a problem now here because directly water cannot be replaced by paraffin or plastic because these two agents, medium, are not able to be dissolved in the water. So because of this, what we do is first we extract water by agents that are able to dissolve in water. Then we replace these agents once the agent has removed the water. We remove it by a medium that is able to be replaced by wax or plastic. That's all what the next steps are. So the first step we need to have should be dehydration. Then there is a process known as clearing. Then we have to infiltrate by the medium we are going to infiltrate into the tissue. And then we will have this type of block which is able to be sliced. So after fixation, we have the following steps. The first step is dehydration. As I told you, dehydration is extraction of water. There are several chemicals that are able to extract water, meaning there are several chemicals which are able to dissolve in the water. It's all the process of dilution. Widely used chemical for this purpose is alcohol, like ethanol. Alcohol is able to replace water. How does it do this? By repeated dilution. Like, for example, in the first vial, you can have 50% alcohol. So the tissue fixed can be immersed into 50% alcohol and leave it for about 1 hour. When the hour is ended, you will have the water in the tissue is not pure water. Instead, it is about 50% alcohol. Then put it in a 70% alcohol, and then put it in 100% alcohol. At the end of the day, you will have in your tissue, instead of water, you will have 100% alcohol. The reason why you put first into 50% alcohol is to prevent the tissue. The tissue has to be prevented optimally. Otherwise, if you put it immediately into 100% alcohol, when the tissue is water is extracted suddenly, the tissue can crack. The cells can be damaged. The tissue outline could be damaged. We are playing gently. So in a process known as dehydration, you pass your tissue in a series of increasing concentration of alcohol. So that's what dehydration is. Now, once water is removed by alcohol, again, the alcohol cannot be replaced by molten wax or plastic, because these two are not again able to be dissolved in alcohol. Instead, we have to find another chemical that is able to dissolve in the or able to dissolve the wax or plastic, as well as it is able to exchange with alcohol. We have, for this purpose, different types of chemical, including benzene, chloroform, and the widely used one is named xylene. So xylene is widely used chemical that is able to replace alcohol, and it itself is able to be replaced by plastic or wax. This step is named as clearing. The reason why the step is named as clearing is because after the alcohol is replaced by substance such as xylene or benzene, widely used one is xylene. After we do so, the tissue looks like translucent, as if something has been cleared off. So the step is named as clearing. We name it clearing. Then after clearing, now the xylene present in our tissue can be removed by process of dilution by molten wax. Molten wax is just nothing, I've been telling you repeatedly, is molten candle, because candle is one type of wax. So you put this in an oven because the wax is solid in room temperature. The other ones could be done in room temperature, but this one should be done in an oven. So the wax is liquid when the temperature is raised to about 60 degrees centigrade. So you put it in an oven and leave it for several changes. After several changes, you'll have your tissue full of molten wax. Now, if you take the tissue out into room temperature, it gets solidified. It would become just something like a candle, which can be sliced by special type of cutting machine. So that's what is known as infiltration or impregnation. After you do this, you take it out. But when you take it out, you have to put it in a mold, just like they make a candle in a mold. So that molding should be in a way that the tissue is oriented for later slicing. I will show you later. The plane of sectioning would matter for later observation of your tissue. So you have to be able to orient like you want, as well as to be manipulated, to be fitted into the cutting machine. So that process is named as embedding. So that's what is named as what we have in tissue processing. This is for light microscopy. This is how we prepared the slides we are going to show you in our histology lab. But if it was for electron microscopy, I told you for electron microscopy, paraffin cannot give us very thin section of about nanometer thickness. We said we need about 40, 50 nanometer for electron microscopy. For that, we have to embed our tissue, infiltrate our tissue by plastic. And that is able to be done by mixing different plastics. So once you cleared, you can put your tissue in one type of plastic. That plastic would be infiltrated because it is liquid. But later, when you mix it with another chemical, it would get solidified. So that solidified tissue is full of that plastic, which is able to be manipulated and which is able to be sliced by a cutting machine. So that's what the tissue processing for microscopy is. Like, for example, here you can see this picture is showing where you can fit your tissue block into a cutting machine. The cutting machine is named as microtome by this microtome, which also has a place for a knife. And by this knife, by rotating a handle, you can cut your tissue into slices. And these slices could be picked onto a microscopic slide, and that's how we slice our tissue. That's the important steps of tissue preparation. These are all listed or summarized in such slides. Here, for example, this slide is showing what embedding is. Embedding is just making your tissue surrounded by the medium you chose. For light microscopy, by paraffin. For electron microscopy, by plastic, where this block could be picked and be put onto a cutting machine, and that machine would slice the tissue to the desired thickness. For light microscopy, the medium of choice is paraffin wax. For electron microscopy, EM means electron microscopy, LM means light microscopy. So for electron microscopy, it is plastic resin. For light microscopy, it is paraffin. You have to also know that the widely used agents for the different steps of tissue preparation, like, for example, I told you, for fixation, it is formaldehyde. Formaldehyde is just nothing, but it is formalin. Formalin is a liquid. Formaldehyde is a gas of same chemical. For dehydration, I told you the widely used one is ethanol. For infiltration and making tissue block, widely used one for light microscopy is paraffin. For electron microscopy is plastic resin. So that's how we prepare. Here you can see the machine for cutting, which is known as microtome, for a process of microtomy. Here is where our tissue block is fitted. Here is a knife, and there are regions where you can adjust the thickness. The thickness could be adjusted. And this is, for example, a microtome known as rotary, because when you rotate it, you can bring this tissue against the knife, and the knife would make the slices. This one is for electron microscopy, where the slices are very thin, which are not as such able to be visualized by naked eyes. So here you have a magnifying glass fitted to this microtome. As I said, for electron microscopy, the tissue thickness should be about 40 to 70 nanometer, whereas it is about 5 to 10 micrometer. I've already told you what micrometers are, what nanometers are. These are 1 millions, these ones are billions. So that's how we take our tissues to different steps for purpose of tissue block preparation for slicing. Again, as I told you earlier, while we are preparing our tissue block, we have to orient our tissue according to later required orientation of your section. Or this is also important for interpretation of the histological slices or photographs you see in histology lab. Like, for example, this is an orange. When you cut it, not orange, it is an egg. This egg in histology lab under microscope could appear any one of this. You may be provided this, you may be provided with this, this one, or any one of these. These are all different sections or different slides from same egg. Their difference is only on the plane of sectioning. So you see, appearance of tissue and organ sections under the microscope depends on the plane of sectioning. So when you look at histology slides under the microscope, you have to also have in mind how these were cut, or the plane of their section. I will show you another more slides like this one, for example. Look at this. This is same bone. It is just one bone. If you slice it or if you cut it longitudinally along this plane, you will get it appear in histology lab like this. If you cut it in cross section, it could appear like a ring. If you cut it obliquely, it can appear an oblique. So these three planes of sectioning are important. So the tissue, maybe the organ, may be cut longitudinally, it could be cut transversely or cross-sectionally, it could also be cut obliquely. And all this matters for the appearance of your tissue under the microscope. Mind you, to show you and teach you about bone, in histology, we are not going to bring this bone to you. We are only to bring to you any one of these three slices. So you have to know how these were cut. So you see, appearance of tissue under the microscope depends on the plane of sectioning. So you have to know how these were prepared. Another slice. Look at this. This is just one same organ, which is coiled. We have this type of organ in our body, blood vessels, for example, also may not be coiled like this. Intestine. One good example, much better than all this, is our sweat gland. Our sweat gland, found in our skin, appears like this. It is more coiled. So in one section, which may cut this coiled tubular, one organ along this plane could give you, look at this, several tubular cross-section, tubular structures. So you see all this, which appear different and many and of different sizes and the shape, may belong to sections of one same organ. So you have to have also this type in mind when you come to histology lab, looking at slides under the microscope. So again, appearance of tissue depends on the plane of section, as well as depends on the gross structure of an organ. That's very much important thing to have in mind. Are you okay with me? I have now finished how we prepare tissue slices for histology study. Okay, thank you very much. Okay, I'll see you are okay. Okay, let me now take you through the next step, because our tissues, unless we impart some artificial contrast coloring, most of our tissue slices are translucent. They appear like this, where you may not able to distinguish the nucleus from cytoplasm. Within cytoplasm, you may not able to distinguish different components we have within our cell. So for this purpose, we have what is known as staining. Staining is just nothing, making contrast or imparting contrast to our tissue. It is artificial. We are making artificial coloring. This type of colorings are not present in our tissue. Instead, we are making some artificial color. And many of the stainings we use for histology depends on the known acid-base type of chemical reactions. Because some of our tissue components have a relatively larger amount of acidity charges, whereas some others have a basic type of charges. So for this purpose, we can have an acidic type of colorful substance or basic type of colorful substances. We call this basic and acidic dyes. Meaning the basic ones, if you pour into your tissue a basic colorful chemical into your tissue, this chemical would be coat because they react with acidic components of your tissue, making the acidic components of your tissue appear like the basic dyes, because many of the basic dyes have their own type of coloring. Same is true for those which are able to react with acidic dyes. So that's what we are going to deal with. Meaning, here look at this. Basic dyes, basic colorful substances, react with the acidic components of your tissue. This type of acidic components of your tissue love or react with basic dyes, so we name them basophilic components. So they are basophilic. What are they? They are acid. They have a large amount of negative charges. And this includes, for example, phosphate groups of nucleic acids. We have large amount of nucleic acid, for example, in the nucleus. Nucleus has DNA, it prepares RNA. This have large amount of nucleic acid, where we have large amount of phosphate, and this acts like negative charges, so they are able to react with your basic dyes. There are also sulfate groups of some ground substance. In ground substance, we have substance known as glycosaminoglycans. These have large amount of sulfate. Carboxyl groups are there. These are all negatively charged. Proteins have also have a large amount of negatively charged carboxyl groups. And this type of tissue components are able to react with basic dyes. Usually basic dyes are colored blue. We have plenty of basic dyes of choices. Many of these come in different shades of blue colors, like this one. Look at this. The first slide, this one, was not clear. It is not clear. But if you stain your tissue by basic colorful substance, it can appear like this, where you can see the nucleus clearly. Some tissue components within cytoplasm are now visible. Then you counter stain this after staining with one type of dye with another one. Earlier we stained it by basic dye. Now you can stain it, same tissue can be stained by acidic dye, meaning you stain for about 20 minutes, 30 minutes by a basic dye, and then remove it, dip it in a water. When you dip it, basic dyes which are not reacted, excess ones, will be removed by dipping it in water. And then you counter stain it with acidic dyes, which would react with basic components of your tissue, which themselves are acidophilic in nature. And these are those containing large amount of, for example, ionized amino groups. Like, for example, most cytoplasmic filaments in muscle, we have such large amount of filaments. We also have in cytoplasm plenty of filaments we are going to see next week. We have like microfilaments are there, intermediate filaments are there. They are all able to be stained by these acidic dyes. Most intracellular membranous components, membranous type of organelles, are able to be stained because they are acidophilic. most extracellular fibers. When you say extracellular, between cells, we have some structures which are, along with cells, give us tissues. So this have plenty of fibers like collagens. They are able to be stained by acidic dyes. So after staining with such dyes, look at this. You can have such beautiful tissue sections. This is, for example, a section of skeletal muscle where you can see one cell. This is one cell. Here is another cell. This one is another cell. Within this cell, look at this is a nucleus. This region is a nucleus. This reddish outline is showing the cytoplasmic content. Between one cell and another, you have additional tissues, additional tissue components, additional structures like blood vessels, like loose connective tissue, are also stained differently because they have different amount of acidity or basic components. The way they would react with same dye is different. They are reacting differently. So the color we get at the end of the day will be different shades of different type of coloring. Usually for acidic type of dyes, we have red, orange type of coloring. For basic type of dyes, we have blue-black type of colorings. So that's what stainings are. And when we come to our day-to-day tissue preparation, tissue staining, we also have one group of chemicals of choices. Like I told you earlier, we have formalin for fixation, we have ethanol for dehydration, we have xylene for clearing, we have paraffin wax for light microscopic tissue embedding. For staining of light microscopy, we have what is known as general stainings. And in a few cases, we also have what are known as group of stains named as special stainings. But the widely used ones, which come under general tissue staining, are two types of chemicals. One chemical is known as hematoxylin, another one is known as eosin. These are the two widely used chemicals or dyes for light microscopy. Hematoxylin behaves like a basic dye. So it stains the basophilic components of our tissue, like the nucleus. Look at this. All these nuclei, this outline of the nuclei, this outline, the round ones, which are stained towards about blue-black, different shades, not just simply blue, not just simply blue-black. It is a different shades. It all deals with the charges, the state of the chemical, the acidity is the matter. So because of this, you have different shades of this type of outline is stained because of hematoxylin. And this would be the tissue would followed by staining with a chemical known as or stain known as eosin, which is acting like an acidic dye, which would mark the acidophilic tissue components like pinkish, orange, red type of outline. So you see, after staining, counter staining your tissue by eosin, you will have this type of sections. Look at this. This is a very beautiful slice or picture of skeletal muscle. This is a very beautiful section of our intestine, where you have different cells stained, different glands are also, outline of these glands are also visible. So this is what our tissue appears after staining with general stains. But mind you here again, any method is not 100% perfect. You see, even following the most expensive and most complex type of tissue processing, you will not have 100% tissue or lifelike tissue structure. there are some changes. There are some which are not able to be visualized, or there are something that would appear because of your tissue processing. So it is not 100% perfect. There are always some which are not able to be visualized, or there are some minor changes. So what we do in tissue processing is always to make our tissue appear as much as like in reality. Otherwise, it's not 100% perfect. Like here, look at this. This region, there was some tissue components here. This is one cell. We name it goblet cell. You will study it when you study epithelial tissue and glands. This cell is also here. It is also here. This region, which looks like empty, is because its component has been washed out. During the tissue processing, while we were fixing it, the tissue component, the cellular component, has been washed away. But if we need, we can retain it. Or this region is not stained because it cannot be stained by hematoxylin and eosin. But if we need, we can stain it by using a special technique. But such a special technique, when we use, this may be not suitable for the remaining ones. Maybe this special technique may be expensive. Maybe it is very difficult. Otherwise, we also have, in addition to general tissue stains and the general tissue preparation, we also have special tissue stains and special tissue preparation. You have to have this in mind. So the method I have been lecturing so far and also continue is just the widely used one, which is just suitable for teaching purposes, which is also suitable just to show us just adequately tissue structures. Otherwise, we have special techniques which can also stain this. We also have some slides which are prepared in such special techniques. So what I would advise you, or what you should do always, is that when you look at the tissue structures under the microscope, you have to know how the tissue is prepared, the way it was sectioned, the type of stainings used. Otherwise, you can see something appearing differently when you look at it after preparing the same tissue by different method. So that's what I can tell you about tissue preparation. And then finally, we have to know a few things about how to look at this prepared tissue, meaning microscopy. So I'm going to take you in a next few slides, microscopy. We use microscope for two important purposes. I know if I didn't show you these two use of a microscope, you may frequently ignore. People, students, always just look at the magnification, think of the magnification function of a microscope. But the microscope has also need also to have a function of resolution. Otherwise, magnification is not the only thing we need from microscope. Like, for example, you can have your picture under, after taking your picture by a camera, you can have it blown up, but out of focus. If it is out of focus, that magnified picture of you is useless. So that picture should also have to show your detail. The detail is the function, it is because of resolution. So a microscope has to have magnification as well as resolution. Magnification makes your tissue artificially, makes your tissue appear bigger and closer to your eyes. So that's what magnification is. Magnification, I repeat, is making your tissue appear wrongly. It is cheating. It is a way, in quotation, it is just making your tissue appear not like the real, but artificially. Makes it appear bigger as well as closer. And resolution is making two separate things appear to you remain separate. That's what the details means. When some objects are smaller in size and are closer to each other, there is a limit. Our eyes able to see them separately. After some cutoff, these two objects may appear to your eyes as if they are just one. So there is a limit, and a microscope has a higher resolution power. So even the quality of a microscope depends on the quality of its resolution power. And for these purposes, for magnification and resolution, a microscope has to have the following basic requirements, components. One is light source. The second are series of lenses. Like this is an old microscope. It has somewhere a cable which would connect it to a electric power source for light, and then different lenses. This type of microscope is named as monocular microscope. This type of microscope, I don't have it here. We also have another ones which are named as binocular. The lenses we have are different. There are different types, and we at least need the following three types of lenses. One type of lens is named as condenser lens. The second one is named as objective lens. The third group of lenses are named as ocular lens. What condenser lens does is it focuses the light generated from the light source onto the specimen. You know, the slide you prepared and stained should come and be put on a mechanical stage somewhere here. And from below, a light has to cross it. And for this light to cross it, it has to be focused. It is like a torch. It is like a torch. You are focusing onto the specimen. So that's the function of condenser lens. There is no function for it of magnification, no resolution. The function of condenser lens, as the name indicates, is to condense, to focus the light upon the specimen, where the light should cross the specimen and form image in the objective lens. So the objective lens is put above the specimen. We need to have choices here. Several objective lenses should be available. So a microscope should have to have at least about 3 objective lenses of different powers. The powers are for magnification. We always begin with an objective lens with less magnifying power for purpose of looking at the specimen with a wider field of vision. So once you look at your specimen in a glance, you spot a region of your interest, and that region of interest should be blown up by using another objective lens with a higher magnifying power. You may begin with an objective lens with a magnifying power of 4. You can change it to 10, you can change it to 20, or you can even take it to 40 and 100. So above the specimen, there should be a place where you can fit, like a rotating turret, you can fit several lenses, objective lenses, and by rotating it, you can change, because the light, after passing through the specimen, is always located here. So you have to bring the objective lens to it. So these are all objective lenses. The objective lenses have 2 functions. The first one is magnification. The second one is resolution. So objective lens is the lens with both functions of the microscope, meaning magnification and resolution. So that's what it does. And when you look at through the objective lens forms image from the object, from the specimen, from the real specimen. It forms somewhere within the tube, a magnified image. This image would then be used as an object for the next lens. The next lens is named as ocular lens. This can come as one in monocular microscope or can come as two in binocular microscope. The modern light microscopy are all binocular, so this type of microscope is what you are going to have. So the function of this ocular lens is to further magnify the image formed by any one of these objective lenses. By any one of these objective lens, you will have somewhere in the tube of the microscope here a magnified image. And this image would be visible, be looked by this ocular microscope lens for formation of a second image. And the function of ocular lens is only to magnify, meaning the image that is produced by objective lens is only magnified. It is unlike the objective lens. The objective lens has two functions. It also would show you the detail. So if I ask you which lens should be expensive, there is no doubt that you say it is objective lens. Indeed, the quality of a microscope depends on its objective lenses. So that's the lenses. And then final magnification therefore depends on the product of magnifying power of the objective lens you used and the ocular lens fitted onto your microscope. The microscope always comes with a fixed ocular lens of your choice. It could be 10 times magnifying ocular lens. It could be 15, it could be 20. Usually, these are the three types of ocular lens magnifying power. It could be about 10 times, it could be 15, it could be 20. Whereas, if this one is, say, 10 times magnifying ocular lens fitted to the microscope provided, you may use one objective lens. When you use the 4 times objective lens, the final magnification would be 4 times 10, 40. When you change it to 20, it could be 20, it would be 20 times 10, 200. If you are using the 10 times, it will be 10 times 10, 100 times. That's how you calculate your magnification of the image. In some modern microscope, there are also multiple images to be created within the microscope. You don't see the lenses within the tube of the microscope or within the objective lens. There are multiple lenses, glasses fitted. You don't see them. If that is the case, they would tell you, and they will tell you about a factor, and you can also multiply it with a factor. It usually comes about 1 point something. So if there is 1.5, you have to again multiply the magnification. So that's how you calculate magnifying power of the microscope you use. So this is what I can tell you about in my first part of the lecture for the day, under Introduction to the Study of Histology and its method of study. So if you have followed me, I am sure you can understand by looking at the slides again or listening to what you have recorded. But this, I am telling you, I can tell you for granted that these are not enough. I have tried to do my best to summarize, to minimize as much as possible. Otherwise, under each slide, You have to enrich it. You have to enrich it by your readings. At least I recommend 2 books